Genes are sections of DNA that encode for a particular polypeptide, with each chromosome being able to carry thousands of genes. One or more polypeptides then fold to form 3D structures known as proteins. Proteins are important as they carry out many significant roles in the body, as well as provide the potential for a particular characteristic, such as hair color and eye color. Genes are only visible during cell division, when DNA condenses into chromosomes where genes are found. Genes also play an essential role during protein synthesis, where sections of DNA are duplicated and translated into amino acids which then form polypeptide chains. As genes code for proteins, millions of different proteins can exist in our body at any given time. Some vital proteins that are responsible for our survival and growth are hemoglobin, a transport protein that carries oxygen around to our tissues via red blood cells; hormones, such as the human growth hormone and insulin. Both are chemical messengers that trigger a response in the body in order to elicit a reaction. Motor proteins, which are essential for muscle movement. They control the sliding of filaments during muscle contraction and the movement of cilia in our lungs and digestive system. Keratin, which is a structural protein that contributes to the structure of hair, nails, hooves, and fur. Enzymes that act as catalysts in the human body. They speed up reactions and facilitate chemical reactions, such as the breakdown of foods during digestion. Gene expression is the process given when the information encoded in a gene is used to direct the synthesis of required proteins. The regulation of gene expression is the process of turning genes on and off, depending on when we need them. For example, different cell types require different proteins to be produced, and so the regulation of gene expression ensures that some genes are turned on and contribute directly to protein synthesis. This ultimately affects the phenotype, which is a species' physical appearance and characteristics. Humans have about 20,000 to 25,000 genes in a cell's nucleus, but only about 10,000 genes are active in any given cell to produce certain required proteins depending on the cell's needs. In a human embryo, genetic information is passed down from both the mother and father. 50% of DNA comes from the mom, while 50% comes from the dad. This is because a diploid embryo possesses 46 chromosomes, and 23 chromosomes are passed down from each parent. This occurs from generation to generation, and each offspring is genetically unique due to the millions of possible combinations of the way alleles switch places during crossing over in the metaphase of mitosis or meiosis.
Let's have a look at CRISPR overview. How was gene editing discovered? Well, who would think that the first discovery of gene editing was from observing bacteria? It was fascinating to scientists when bacteria infected by viruses could recover themselves and gained improved immunity. This is because of the built-in genome editing system that allows bacteria to detect a specific mutated DNA sequence and remove it. The removed section is then rebuilt back to normal by the natural recovery process of the cell, or add with a specific DNA section. Scientists studied this genome editing system and applied it in many fields, including improving the immunity of animals in agriculture, studying the drug resistance of some bacteria, and treating diseases involving genetic materials. So, what is CRISPR? CRISPR, or clustered regularly interspaced short palindromic repeat, is one of the tools used in gene editing. The system consists of mainly two parts attached, a guide called gRNA and a DNA cutting enzyme. A gRNA is a strand of RNA consisting of approximately 20 nitrogenous bases. This is to match the nitrogenous bases of a targeted section of DNA and bind to it. A DNA cutting enzyme is a protein that can cut DNA strands to initiate the genome editing process. So, how does CRISPR work? CRISPR can regulate gene expression and produce functional proteins by changing a nitrogenous base in a specific gene section. The DNA bit removed by CRISPR can be recovered or changed into a specific genotype, resulting in the new expression of protein and thus phenotype.
Parkinson's disease is a neurodegenerative disease that many people around the world suffer from due to the progressive deterioration of their neural function and its untreatable nature. One of the causes of Parkinson's disease is the presence of aggregated proteins called Lewy body, which is made up of several types of protein, including alpha-synuclein, which is encoded by SNCA gene. The mutation of the SNCA gene at the LR53THR position was found to be the cause of alpha-synuclein aggregation into the Lewy body, and thus causing Parkinson's disease. Yoon conducted an experiment putting A53T SNCA mutated mice into a control group and a treatment group. Only the treatment group was administered with CRISPR. To measure the result, rat behavioral tests including stepping and cylinder tests were used to assess their neural function. Alpha-synuclein level was used to determine the presence of Lewy body and Parkinson's disease. The results found that the treatment group showed better performance in the rat behavioral tests and had a lower alpha-synuclein level, leading to the conclusion that CRISPR is a potential approach to prevent a missense mutation-derived Parkinson's disease.
Another CRISPR application in medicine is lowering cholesterol. Cholesterol is good, useful, and delicious. We just love it. However, having too much can make your blood vessel lose their electricity, possibly leading to cardiovascular disease and stroke. Lucky us, on the surface of our liver cells have the LDL receptors. These receptors help to reduce bad cholesterol called LDL by binding to LDLs, bringing them to the cell, breaking them down before storing or removing them from the body. The number of LDL receptors is regulated by a protein called protein convertase subtissue-linkexin-9, or PCSK9. It was found that the mutation of PCSK9 gene caused overexpression of PCSK9 and reduced LDL receptor number, resulting in high blood cholesterol. A study published by Nature 2021 was a new study to the efficacy of CRISPR in preventing the overexpression of PCSK9 and reducing LDL in monkeys. They mutate PCSK9 gene in monkeys and put them into control and treatment group. Treatment group received CRISPR to modify the mutate PCSK9 gene, and the control group only received placebo. To evaluate the outcome, blood tests were performed to measure blood PCSK9 and LDL levels, and the level of enzymes released when the liver is damaged, which are alanine transaminase or ALT, and the aspartate transaminase or AST. The result shows that monkeys who receive CRISPR have 90% lower blood PCSK9 and lower blood LDL levels equivalent to the efficacy of cholesterol drugs, and also no sign of liver damage as indicated by low ALT and AST levels. Now from this study, we can conclude that CRISPR can potentially be a once-and-done treatment to high blood cholesterol and outshine cholesterol drugs in terms of the safety and of the liver and the quality of life, as there is no need for lifelong drug consumption to maintain an appropriate level of blood LDL.
Increased applications in the field of infectious diseases have resulted from a better knowledge of CRISPR-Cas biology. CRISPR technology promises to clarify fundamental host-microbe interactions, aid in the creation of speedy and accurate diagnostics, and advance infectious diseases prevention and treatment. Recent CRISPR applications for the creation of infectious diseases diagnostics take advantage of Cas12 and Cas13 nucleus-induced collateral cleavage. Specific high-sensitive enzymatic reporter unlocking is the first of these technologies, which combines isothermal recombined polymerase affrication or reverse transcription (RPA) with Cas13A cleavage. With great specificity, a cRNA Cas13A complex binds and cleaves target nucleic acid. When the cRNA's Cas13A complex attached to the target nucleic acid, non-target RNA connects to the fluorescent reporter is also cleaved, resulting in a fluorescent signal for pathogen detection. The capacity to distinguish single nucleic alterations is one of the features that makes this technique appealing. First CRISPR-based therapeutic trial in the United States combined CAR-T and PD-1 immunotherapy techniques, editing 3 genes with CRISPR. The Parker Institute and the University of Pennsylvania collaborated on this phase of 1 study, which began recruiting in 2018 and ended February 2020. The purpose was not to cure patients, but to see if the treatment was safe and had tolerable adverse effects. 2 patients volunteered or treated, one with advanced white blood cell cancer and another with metastatic bone cancer. It was found that the therapy was simple to use and had minor negative effects. The T cells settled in a month. T cells were able to detect and infiltrate tumors. According to tumor biopsies, off-target impacts were only seen in a few cases. During manufacture, however, undesired mutation at the target site was common, with 70% of cells revealing at least 1 mutation at or near the target location. The percentage of cells with mutations decreased in patients after infusion throughout time. These findings suggest that CRISPR-engineered CAR-T therapy could be a promising treatment option. It's safe, it has manageable side effects, and does not trigger a robust immune response.
A major concern with using CRISPR-Cas9 for gene therapy is a high frequency of off-target effects. Off-target effects are serious as they may cause loss-of-function mutations in proper functional genes or incorrect repairing of disease-inducing genes due to binding and breaking at sites other than the target DNA sequence. Furthermore, it may also lead to chromosomal rearrangements and other types of mutations. Current methods to minimize these occurrences include the use of Cas9 nickase with an sgRNA pair targeting both strands of the DNA at the intended location. Researchers have also developed specifically engineered Cas9 variants that reduce off-target effects while maintaining efficacy. The ethics regarding the safety of gene editing on humans encompasses 2 categories: genetic modifications to correct faulty genes and genetic enhancement. While gene therapy with somatic cells is generally considered ethically sound as they only affect the patient and is not passed down to future generations, the genetic enhancement of these cells as well as the genome editing of germline cells is widely met with controversy. The concerns of germline editing extend into 2 concepts. While on one hand, scientists may be able to prevent genetic diseases from being spread to later generations, the risks involved are a cause for concern. Until these risks are dealt with and the overall safety of the treatment is improved, editing of germline cells remains a highly debatable topic. Probably one of the most controversial concerns is the use of genome editing to genetically enhance individuals. If done with germline cells, scientists could potentially practice eugenics to create superhumans by increasing strength or endurance. Already, CRISPR-Cas9 edited pigs have been designed to be resistant to viral infections, possess improved thermogenic capacity, and accrue less fat mass. CRISPR-Cas9 engineered beagles have also been designed to have double the muscle mass of their non-engineered counterparts. In conclusion, genes play a significant role in our body as they encode for many different proteins which are necessary for survival and growth. With the latest development of CRISPR, we can now edit any organism's genome to produce breeds with specific characteristics and traits to improve daily life, such as selective breeding in the agriculture industry to boost productivity. Medical applications of CRISPR can also bring about potential cures for diseases such as Parkinson's disease and cancer immunotherapy. However, the CRISPR technology has raised ethical issues within society as well as caused increasing concern over the off-target effects which can lead to unforeseeable risks. Hence, more research and trials must be performed in order to increase the efficiency and accuracy of this promising technology.